Dr. Barber, please. Thank you.
All sorts of strange contraptions on my head. Anyway, thank you very much. I'd like to thank Esper and et al. For the kind invitation to come to Sao Paulo. It's always a pleasure to come to Brazil. And today I'm going to talk mainly a little bit about my work and our contributions to the field of innate immunity and a little bit of HIV, but I don't really work on HIV that much. I'm not clever enough. So let's see if it works. So hopefully this will introduce you to something other than a little bit of HIV. So I'm interested in the production of interferon and cytokines. So my interest lies in understanding how viruses stimulate the immune system. And a key mechanism of following virus infection is the cell responds by producing many, many cytokines, chemokines, and materials referred to as interferons. The interferons are triggered by virus infection. There are about 148 amino acids in length, and following the virus infection, interferons produced, and then it binds to receptors on uninfected cells, and through JAK-STAT signaling, produces hundreds of antiviral genes. And the interferons, there's one interferon beta gene, about 12 interferon alpha genes, intronless. These are secreted, and they stimulate antiviral, they make antimicrobial proteins to stop the virus from replicating. They also stimulate the adaptive immune response, T cell and B cells. The innate immune response is critical. If you don't have any innate immunity, the body doesn't have time to generate B and T cell responses, and you die. So the innate immune response is really very important until the time you generate B cells and T cells. So the appropriate induction of interferon is critically important, but through studying these processes, we've also learned that the inappropriate triggering of these mechanisms can lead to inflammatory disease. Oops. Sorry. Let's see. Okay, sorry. So anyway, how are the interferons made? Well, the virus infects the cell, and then somehow the virus triggers activation of transcription factors. The transcription factors enter the cell, enter the nucleus, bind to the promoter of interferon beta and alpha and many other genes, trigger the production of interferon, binds to other cells, and induces hundreds of genes. And that's how interferon works. So we knew a lot about transcription factors, and we knew a lot of how interferon produces the proteins, but what was unknown was what's the first sensors are. What are the first sensors which trigger the production of interferon? So the interferons were discovered over 50 years ago, but only in the last 10 years do we understand what those sensors are, the important sensors for triggering our host defense response. And it became apparent that in relation to viruses, that there's a toll receptor pathway, TLR3, toll receptor like 3, recognizes double-stranded RNA species, virus species. TLR7, predominantly expressed in plasmacytoid dendritic cells, binds to TLR7 in single-stranded RNA. And TLR9 recognizes single nucleotides of DNA, 21 bases, 21 nucleotides, CG-rich. And so the toll pathway is important for recognizing RNA virus. And then about 2004, Fujita and Yoneyama discovered these helicases called RIG-INDA5. And these are critically also important for recognizing viral RNA. So the most important receptors for recognizing RNA viruses are TLR7, TLR3, and the RIG-I pathway. And between them, these receptors are responsible for recognizing all RNA viruses and triggering the production of interferon. So we pretty much know now what the sensors are, only in the last decade. But what was unclear is how do DNA viruses or retroviruses activate interferon? And people still didn't know this until around 2008. So what are the major triggers? For example, herpes simplex virus, bacteria, malaria, parasites, maybe retroviruses that have DNA intermediate genomes. And this is where my own laboratory came in 2008, and we were looking for proteins or cellular proteins which activate the production of interferon. And we screened about 18,000 clones and found one gene that when we overexpressed it in a cell was potently able to trigger the production of interferon. We didn't know much about this protein. It was an unknown protein. We called it STING, for stimulator of interferon genes, because all we knew is that overexpression of this generated interferon. So this STING molecule seems to reside in the endoplasmic reticulum. It's in all hemopoietic cells, macrophages, dendritic cells, etc., and also in epithelial cells, small airway epithelial cells, endothelial cells. STING is present in cells that are exposed to the environment, as well as in immune-related cells. And it became clear what we did actually was make a mouse which was deficient in STING. We generated the mouse, and it was viable, it was alive. And what happened was we were able to generate murine embryonic fibroblasts from the mouse, or take macrophages from the animal. And then what we did was able to stimulate the cells by treating them with known activators of interferon. So what can activate or trigger interferon production is DNA and RNA. Okay, so double-stranded DNA species or single-stranded RNA or poly-IC or synthetic nucleotides are very good at producing interferon because the toll pathway in Rig-I recognize RNA moieties. They recognize the RNA genome of the virus. So if we take RNA, poly-IC or double-stranded DNA and transfect it into a cell, the cell responds by making interferon like this. This is interferon beta here. This is a macrophage, this is a dendritic cell. So the cell responds when we put in DNA, or we put in a bacteria or a DNA virus, we infect the cell or treat it with DNA, we get lots of interferon produced. However, STING knockout cells, there was no production in the green, no production of interferon in response to HSV, listeria, double-stranded DNA species or anything. So cells lacking STING were totally unreceptive to DNA, the production of interferon by cytosolic DNA. This is what happens if you take a virus, like an RNA virus, but in this case a DNA virus, herpes simplex virus, and you place it onto a macrophage, for example. So you have a HSV, and you put it onto a macrophage, and the cell responds by stimulating all these hundreds of genes. This is a heat map. Chemokines, CLCX10, etc. But STING knockout, STING deficient macrophage, there's nothing going on at all. So we rapidly became clear that STING was very important for the recognition of DNA pathogens and for the recognition of production of interferon in response to DNA species here. This just goes to show you that when we take the poor mice and we infect them with herpes simplex virus, they die very, very quickly because they can't produce interferon. So STING-deficient animals are extremely sensitive to DNA pathogens, including bacteria. So I've just mentioned STINGs in the cytosol here. And then in the endoplasmic reticulum. And then when it sees something like DNA or a virus or a bacteria, what happens is STING undergoes this strange trafficking event, which is a form of autophagy. So it moves when it produces interferon. It traffics in response here. So we want to know what's upstream of STING and what's downstream. How does this process function? How does the DNA viruses or DNA or the bacteria produce interferon? Well, we discovered STING in 2008. In about 2011, this guy, Dan Portnoy from California, noted that bacteria could trigger the production of interferon. And it did this by generating these little cyclic dinucleotides. Cyclic DAMP, secreted by intracellular bacteria, can trigger interferon. He noted this. It turns out that these cyclic dinucleotides, which look like this here, trigger STING by actually binding directly to it. STING is a dimer in the endoplasmic reticulum. And these cyclic dinucleotides generated from bacteria can bind to STING, and then STING undergoes this trafficking event here, It moves very, very fast. And so cyclic dinucleotides, so we've evolved, created a protein that's able to recognize cyclic dinucleotides generated from bacteria. Then the Chen laboratory found out that Sting was able to recognize DNA viruses by the DNA from the pathogen binding directly to a cellular protein called cGAS, cGMP synthase. So in the body, we have this molecule of 522 amino acids, and it recognizes DNA from DNA pathogens, and then makes these little cyclic dinucleotides. It's an enzyme which creates them. And these enzymes then bind to Sting. So we have a direct method. Sting is a direct sensor for cyclic dinucleotides generated by bacteria or generated by cGAS following the binding of DNA here. So cyclic dinucleotides able to bind Sting to produce cytokines. We have this very strange trafficking event here. It turns out the Sting is part of the translocon complex. And this is a kind of a hole in the endoplasmic reticulum. And what happens is all proteins that are glycosylated, N-glycosylated, or secreted, have to be put into the endoplasmic reticulum through this translocon complex. And this is where N-linked glycosylation occurs. So the proteins, about 30% of all our proteins are either glycosylated or secreted. So this is an important organelle. Proteins come down here and glycosylated and then secreted. So Sting is part of this translocon complex. We don't understand why yet. So this is what we think is happening. Sting is in the endoplasmic reticulum, and then it undergoes trafficking in a form of autophagy. So what happens is Sting is activated. We don't understand what triggers its movement. There's a lot of phosphorylation events and a lot of ubiquitination events occur. And then Sting carries TBK1 in these regions to fuse with endosome carrying NF-kappa B and IRF3. Then TBK1 phosphorylates these and activates cytokine production. So it's still a lot we don't know about this process here, but the trafficking is very important for Sting function. And Sting meets transcription factors here. Following this, Sting is rapidly degraded, completely degraded. The reason it's degraded is because we do not want sustained cytokine production. And I'm going to tell you why. So this is a cartoon just summarizing this that we brought out last year. This is Sting in the endoplasmic reticulum. Bacteria makes cyclic dinucleotides, binds to Sting. Sting carries TBK1 to these regions, and then the transcription factors are activated. Or cyclic dinucleotides generated directly from bacteria can bind Sting, or DNA from pathogens bind to cGAS, which generate these cyclic dinucleotides. So this has only been discovered in the last few years, but it's a very important component of innate, how we recognize pathogens. And it's only just been unraveled in the last few years. So the production of interferons are very important. So in addition to production of interferon here from pathogens, the production of interferon is also essential for the generation of adaptive immunity, for the stimulation of T cells, and interferon production and interferon function on subsets of D cells and antigen-presenting cells. And if you don't get interferon produced via Sting signaling, you don't get effective anti-pathogen T cells or anti-tumor T cells, and I'll talk to you about that in a bit. Now, what's a little bit of interest from a translational perspective is that there are many groups looking for activators because Sting activators have proven to be very good adjuvants for the stimulation of immune responses, for example, in vaccine formulations, etc. So there's going to be a new spate of interest in utilizing Sting agonists for generating new vaccine concepts. So for those interested in infectious disease, when we first found Sting, we noted that it also had very close homology to a subset NS4B of yellow fever and dengue virus. We're still not quite clear, but we think that these two viruses, yellow fever and dengue, inhibit Sting function in order to avoid pathogenesis. And we're still working on this, but there's very, very close homology to a certain portion of Sting with yellow fever and with dengue virus infection. Relating to HIV infection, there's been literally dozens of manuscripts now published on this. Obviously, you're very aware of this, but the RNA and DNA form here. There's ample opportunity for HIV and other forms of retroviruses to be recognized. And when the RNA to DNA form occurs from HIV and the DNA enters the nucleus, we think that this part here may activate the sting, may be triggered at this point here. I won't go into it all, but you can read. There's many, many papers in a lot of high-profile journals which have looked at the role of sting signaling in HIV pathogenesis. And you can follow some of this work here, and there's a review actually out last year which I can refer you to. So again, this is a summary of what goes on, that HIV may activate C-gas through this, but also has found a way of mechanisms of maybe suppressing it. Not many people have done or looked at sting function in T cells yet, believe it or not, in any depth, but sting is present in quiescent T cells. So, why doesn't sting or other innate immune pathways clear HIV infection? And I think, as I've just mentioned here, these are just some of the reasons that it might be that you can read about in the reviews. This, again, is which is the review I wanted to refer you to, Nature Immunology, which gives a comprehensive evaluation of what's known about or postulated to occur relating to sting function and HIV pathogenesis here. So the appropriate induction of interferon is essential, but I've just mentioned that the inappropriate induction of these very same pathways have led us to understand other forms of disease. For example, autoinflammatory disease here, and I'll talk to you a little bit here. Okay? So inflammatory disease manifests itself in all types of forms: inflammatory bowel disease, rheumatoid arthritis, systemic lupus. Maybe 20% or 30% of the population will suffer some form of inflammatory disease. Inflammation drives cancer, and so on and so forth. But the thing is, not many people understand what causes the inflammation or the cytokine production here. So it turns out that these pathways are very important. I'm just going to show you a couple of slides. It's been known for some time, for example, that patients suffering from a severe form of systemic lupus, the patients generate an awful lot of cytokines in their blood and their body, and they usually die when they're very young. And when people sequence these patients suffering from this disease called Aicardi-Gutierrez syndrome, typified by having lethal encephalitis characterized by high levels of interferon in the bloodstream. Turns out they had a mutation in an exonuclease referred to as Trex, DNase 2 or Trex1. So all the main mutations were just in that one gene. These patients didn't suffer from infection or anything like that. Quite the contrary, they had lots of immune responses, but they usually died when they're born. Well, it turns out that there's a mouse that also has a defect in this Trex gene, and the mice suffering from loss of this exonuclease die also. They have a severe form of myocarditis, inflammation-driven myocarditis. And the mice lacking Trex also die around 8 weeks of birth. Anyway, obviously we could be able to cross Trex knockout mice with sting deficiency to evaluate the importance of sting in this. And this is what a Trex lifespan of Trex deficient animals are. They usually start dying very early, but the mean is around 50%, is around 8 weeks. You take a Trex mouse with a sting deficient mouse and cross them, and you're completely normal. The mice are completely healthy. So this disease, or the generation of these cytokines, is through sting, because when we take out sting, these mice are completely normal. So inflammatory disease has now been found to be caused through the sting signaling pathway a lot. And this is just a summary slide. You can read about it if you want. But what we think is happening in the case of Trex1 is this. Following cell division of macrophages, predominantly in immune cells, after cell division, the little pieces of DNA are left and reside in the cytosol. And these exonucleases, for example, Trex1, chew up the DNA that's left over after replication, chew it up. And if it's not chewed up, then it activates C-gas to produce sting-dependent cytokines. So now there's becoming more evident that we understand, through understanding sting-related pathways, we've learned a lot about autoinflammatory disease only in the last few years. And this is a case in the New England Journal of Medicine of actually, I've just shown you before, there's defects in exonucleases that are designed to control sting. There's also cases where we have gain of function, where there's point mutations in sting itself. And what happens in these cases is sting is permanently active. It can't be controlled, and it's permanently making cytokines. This is a less severe form. In more severe forms, which we've isolated from patients, I had a call, it was a patient, quote unquote, the patient turned out to be a 3-month-old baby with one amino acid change in sting. And the patient died before it even reached 4 months old from massive amounts of cytokine production. When we cloned this gene in our hands, it turned out that this one amino acid change in sting rendered sting permanently active and producing cytokines at extremely high and evidently lethal doses. So understanding these pathways, I think the role of sting perhaps in multiple sclerosis, all forms of inflammatory bowel disease, maybe dementia and inflammation, rheumatoid arthritis, etc., and all forms of autoimmune disease may involve these innate immune pathways. I'm just going to finish a little bit with talking about infectious disease. I've talked a little bit about inflammation, and the recent considerable interest in these pathways comes from in the cancer field. And as you know, inflammation, chronic inflammation can drive all forms of types of cancer, especially viruses, hepatitis B and hepatitis C, for example, cigarette smoke, etc. So what happens is inflammation triggers these innate immune signals, and these innate immune signals generate cytokines and growth factors, which constitutively tells the cell to proliferate. And that's how inflammation drives cancer. So it's been known for actually some time that carcinogens and chemotherapeutic reagents, such as cisplatin, etoposide, can cause DNA damage and induce interferon production, but it wasn't clear how. It was also known that TNF-alpha was a big player. So what we can do is you can actually take molecules like this, and you can actually paint mice on the skin, and they develop cancer. And it turns out that if you don't have sting, mice are resistant to inflammation-driven carcinogenesis. I'm not going to show you that data. I'm going to show you something here which shows that carcinogens do activate innate immune pathways. This here is a wild-type cell. This is a sting knockout here. And this is that T-Rex knockout mouse I told you about. And this is a double knockout, sting knockout, T-Rex knockout. The bottom line is you put carcinogens such as etoposide or DMBA, carcinogens, they bind to DNA, cause DNA damage. The DNA comes out of the cytosol from the nucleus. You start to get chromatin restructuring. Then you get cytokines produced. Okay, and this is a sting knockout, there's much less. But the T-Rex one, where there's no T-Rex and this sting is present, you get very high levels of cytokines. It's more typified here. This is when you treat with a carcinogen, the DNA loops out into the cytosol. This DNA activates sting. It's under control a little bit, but in the absence of T-Rex, you can see that cytokine production goes through the roof. As T-Rex is desperately trying to chew up the DNA in the cytosol to stop the cell from becoming too inflammatory. So you can see it very clearly here. Then you use a sting knockout, T-Rex knockout cells, completely goes away. So this is sting-driven inflammation in response to DNA damaging agents here. Sting-deficient mice are resistant to carcinogenesis through inflammation. And this is what we think is happening. You get DNA damaging agents, and again, the DNA comes out of the cytosol, activates, T-Rex is desperately trying to chew it up, exonucleases. If it doesn't, sting gets activated, and you get inflammation. So it turns out the sting that's chronic sting activity. Transiently, as we've seen for viruses, it's very important for protection. It also turns out to be very important for protecting against early stages of anti-tumor T-cell responses. So this is a mouse that's been fed carcinogens. And it turns out in this situation, in this model, that sting knockout, this is massive amounts of inflammation. If you don't have sting, you generate more cancer and more inflammation in the stomach. Because it turns out that sting is essential for early detection of DNA damage. And it makes these wound repair cytokines which come and fix the cells. But if you don't get that, bacteria infiltrate and they cause further inflammation, believe it or not. So sting is going to be essential for protection, generating early immune responses to protect against cancer. And then the last two slides, it turns out that from work on sting knockout mice, found out that sting-deficient animals don't generate very good antiviral T-cell responses, but they also don't generate very good anti-tumor T-cell responses. In this model, you can take a sting knockout mouse and actually inject it with a B16, for example, syngeneic melanoma. These are immunocompetent animals. And what happens is the sting-deficient mice, the tumors grow much faster through a lack of generation of anti-tumor T cells. This is a wild type one here. So sting-dependent cytosolic DNA promotes anti-tumor T cell activity. And people have got a little bit excited about this type of work now, and what's happening is they're taking all sorts of STING agonists and stimulators and cyclic di-nucleotides and injecting them into the tumors directly. It's actually in phase 1 trials now. And these agonists of STING, as I mentioned before, stimulators of STING, are very good at generating anti-pathogen T-cell immune responses, but also very good at stimulating anti-tumor T-cell responses. So the immuno-oncology market is very interested in this approach now, and there's lots of clinical trials going on using STING agonists for cancer, as well as in vaccines. And this is what we think is happening in the last slide. We think we get DNA damage events, and what happens in the case of cancer is the DNA damage alerts STING. STING makes cytokines. This attracts macrophages and antigen-presenting cells. The macrophage eats the tumorigenic cell or the virus-infected cell. And then what happens is STING in the phagocyte, which is activated by the pathogen nucleic acid or by the dead cell DNA, makes cytokines which facilitate cross-presentation and cross-priming. And if you don't have STING in either of these compartments, you don't generate very effective antiviral T-cell immunity. So that's that's a whirlwind tour of STING and infectious disease, and inflammation and cancer. And I think the guy he visited me, this is the guy Ishikawa who found STING. 2008, he's now in Okinawa, and this is the rest of my group here who did a lot of this work. So anyway, I know it's not on HIV too much. I did put 3 or 4 slides in, but I hope it was a little bit informative, and I'll be happy to answer any questions.